Okay, part 3. This is the electron transport chain. And what you all need to know is that there is another name for this. Electron transport chain is what we always teach sophomores in biology. But in AP, this is known as oxidative phosphorylation. And of course what that means is that you lose electrons, and I'll show you where the electrons are going to go, then we're able to put phosphates on things. So it's oxidative phosphorylation. So you all will need to know that term, that one in particular, oxidative phosphorylation. That's what this process is called. Alright, so what you're looking at is the mitochondria. What happened was, let me zoom in on this a little bit. What happened was is that we were last talking about the Krebs cycle. And the deal with that is that we are now in the mitochondria. And what we have here is something called the matrix. That's kind of like the fluid. Instead of saying cytoplasm, this is the cytoplasm of the mitochondria. But so that we don't get it mixed up, we're going to call it the matrix. So the Krebs cycle, or citric acid cycle, took place in here. So here's our NADHs and our FADHs, ATP, CO2, all this stuff that came out of the Krebs cycle floating around in here. Of course, there's hydrogens floating around in here, and there's all kinds of chemical compounds, just like in the cell cytoplasm, that are floating around in there. Because, you know, the cell's just jam-packed with things. And so here's what's going to happen. What you are looking at is, in this picture, a mitochondria, and here are the cristae, and these are a double membrane type of thing. You're going to have inner membranes and outer membranes and the matrix, etc., etc. I drew this humongous one, and those of you that had me for honors already will recognize this drawing. But anyway, what you're looking at are what are called transports. And what the deal is that conformationally speaking, there are receptors on here for certain of these compounds that we made. Example 1, NADH will fit here. And so what happens is that when this touches this breaks, this bond breaks right there. Okay? And when you break that, not only are you going to have NAD+, which is going to go back to the Krebs cycle and do some more work, but you have released hydrogen. But what you let go of were electrons. And so here's our electrons. Okay? FADH2 comes down here and enters there. It's farther down the chain per se. And so what happens is that it's going to release FAD and hydrogen ions. Okay? And then it's also going to put those electrons there. So what we're saying is this is an electron transport chain. And so what we're having happen is that when you put electrons in here, which have a negative charge, they are going to pull protons or hydrogen ions into this inner membrane space. And so you were creating what's called a proton gradient. But that's neither here nor there yet. You also have these intermediate carriers, kind of like in photosynthesis. One of these is actually called CoQ. We'll talk about that in a minute. But anyway, these electrons, once they've pulled the hydrogen ions across, are going to jump or ride over to the next transport. Now when the electrons get to the final transport, they're worn out. They have expended their energy by pulling the hydrogen ions across. And so something has to pick them off. In other words, if these electrons were to stay there, then these can't move in. And so what we have is oxygen sitting here waiting for those electrons. And when oxygen picks them up, it is then able to hook on a couple of hydrogen ions to become water. So oxygen is called the final electron acceptor. And if you'll recognize that, this is why we breathe. Because if you quit breathing, then you're no longer going to make ATP, which is the purpose of everything we're doing. So let's look at it again. NADH starts at the top of this chain, but FADH comes in at the middle of the chain. That's going to be important later. These electrons are there to pull hydrogen ions or protons across into the inner membrane to create a gradient, and then they jump and move down this transport. It's called moving down the electron transport chain. Alright, you have intermediaries there that will help this occur. The reason I mentioned CoQ is because you can buy CoQ10 in the grocery store. And then if you take this molecule, it enables you to more efficiently use the electrons that are being passed off. And it helps you use your oxygen more efficiently, and it should help get rid of cramps and things like that that are caused by lack of oxygen and lactic acid buildup. All right, so we've done all this. We've used our electrons, and what are we going to do now? Well, remember from photosynthesis that these hydrogen ions are going to have to equal out because we have a bunch of hydrogen ions over there and not a whole lot here. So what they're going to do is to come over here to this thing that's called ATP synthase, just like in photosynthesis, which kind of gives you an idea that evolutionarily speaking, synthase. Yeah. We're very similar to plants in that effect. But anyway, these hydrogen ions are going to go down ATP synthase, turn the rotor, and we're going to hook ADP up with some phosphate groups to make ATP. And the deal is that for every 2 hydrogen ions, you're going to get 1 ATP. It's going to be important in a second also. NADH at this point would have made 2 hydrogen ions there, 2 hydrogen ions there, and 2 hydrogen ions there. So that means that the NADH that went in right here could literally make 3 ATP. FADH2 goes in at a different place, so it can only make 2 sets of ATP. So that's going to be important. All right. What you're looking at is the purpose of larger organisms needing to go through 2 more steps to make ATP, because we're going to get a lot more bang for our glucose buck by doing this. You remember in glycolysis, what we had was, I'm sorry if you all can hear things over in the background. Pooty's knocking stuff off the table. All right. What we had in glycolysis was only 2 ATP made. But see, now we're all the way into the 3rd step in larger organisms, so we're going to make a whole bunch more ATP. Let's also talk for a second here about the fact that you have to breathe in oxygen, whereas the other ones in glycolysis didn't need it. When you breathe in oxygen, you're able to siphon off these electrons and keep the system moving more efficiently. And of course, the more oxygen you breathe in, the more ATP you can make if you need it. If you get enough ATP built up, the gradient says stop, and then you'll quit making it for a while until you need some more. All right, so let's look very briefly at what we have done, because this is what you are going to need to know. All right, in glycolysis, you made 2 ATP. You also made 2 NADH. All right, in oxidative decarboxylation, you made 2 NADH. In the Krebs cycle, you made 6 NADH and 2 FADH2s and 2 ATPs. Now you're going to ask me, if you go back and look, well, I only saw you write down 3, 1, and 1. But remember, when we had our original 6-carbon molecule, we had to break it down into 2 3-carbon molecules. So everything is going twice, per se, after this step. All right, now here's the biggie. Really, there's different numbers of ATP that are going to be made depending on where these things enter these transport chains. And it will also depend on where you are and what energy needs you have. So I'm going to put out here our ATP count. So you have 2 ATP here, can't change that, and you have 2 ATP here, can't change that. This NADH is not going to enter that electron transport chain at the same place. And so this NADH, rather than, as I told you, making 3 ATP, is only going to make 2 ATP, and that's a total of 4. This NADH has issues. Sometimes it's 2, sometimes it's 3. Most of the time it's going to be 2, so there's 4 ATP. But the ones that are in the Krebs cycle, which are in the mitochondrial matrix, can make 3 ATP. So that's 18. Remember that you've pulled the 3 sets of hydrogen ions across the transports. And then the FADH2 goes in at a different place, so it only makes 2. When you total this up, 2 and 4 is 6, 6 and 4 is 10, 10 and 18 is 28, and 28 and 6 is 34. However, your book says 36 to 38. What the deal is that if you make more, if this enters where it can make actually 3 ATP, or that one enters where it can make 3 ATP, you can eke out more. Given most books, it will say 36 to 38. So that's the number that you need to remember. So in other words, anaerobic respiration will only get you a net gain of 2 ATP, period. So that's not good for you at all. Great for a yeast, great for a bacteria. Aerobic respiration, in other words, where you are breathing oxygen to pull things on down the line, that literally will, of course, will get you a net gain of 34 to 38 ATP. And that, of course, is more efficient, and evolutionarily speaking, that helps us a whole lot as large organisms.